This tutorial will show you how to use the software BLAST in conjunction with the ABE Saving a Life module. BLAST is a biological database that also functions as a search engine and allows you to compare different types of biological data. If you've used BLAST before, be aware that it was updated to a new version in July of 2021 and thus might look a little different than you're used to. To begin, find the BLAST website. It's easily searchable on Google. For this module, we'll be using Protein BLAST. Enter the series of unknown amino acids into the box in the upper left-hand corner. Scroll down to the bottom and select the Quick BLAST option in the algorithm box. Then click the BLAST button. BLAST will now compare this series of amino acids to its entire database. Because the database is so big, this can take a few seconds, so please be patient. When the results come up, you'll see a list of potential matches. The results will be divided into multiple columns, but there are two that are going to be the most useful for us. The query column will tell us how much of our sample sequence is included in each of these matches, up to a maximum of 100%. The percent identity column will tell us the extent to which the two sequences have the exact same nucleotides or the amino acids in the same positions. As you can see, a number of these proteins that popped up in the results are perfect matches. We can find out more about potential matches by clicking on them. This more detailed view also has the option to show alignment. In short, it stacks two sequences on top of one another and visually identifies the areas in which they correspond and areas where they differ. You can see this by clicking the word Graphics next to the title of each match. If you scroll to the bottom of the results page, you'll notice that the matches are less good. Only some of the amino acids here match up with the sample sequence that we entered into the BLAST search box. As you can see, the alignment here isn't nearly as good as the one we saw earlier. Because the amino acids are not all identical between our query sequence and this potential match, you can see that there are a lot of gaps. By examining the results that come out when you searched for our sample sequence within BLAST, hopefully you will be able to identify each of the mystery proteins.